Metabolism refers to all the chemical reactions occurring within the body. Enzymes are biological catalysts which increase the rate of chemical reactions. The enzymes support the metabolism to keep it up to speed, capable of sustaining a life form and maintaining proper metabolic functions in an organism. Enzymes' efficiency can be affected by certain factors including temperature, pH and substrate concentration. Optimum enzyme function will operate efficiently within a limited range. Therefore, a constant and stable internal environment is needed so that enzymes will be able to work at an optimum rate, and thus metabolism will be an optimum efficiency.
Most enzymes are made up of proteins. Proteins consist of one or more polypeptide chains. A polypeptide chain is an amino acid chain connected by peptide bonds.
Part of the enzyme is called the active site. This part attaches to the substrate. The substrate are molecules the enzyme acts upon. Enzymes are highly specific. Each enzyme acts upon one substrate only. This is because the shape of the active site in the enzyme matches the shape of the substrate.
Enzymes perform the critical task of lowering a reaction's activation energy. Two substrates can be joined, or one substrate can be split.
The lock and key model suggests that the substrate fits exactly into the active site of the enzyme like a key fits into a lock. It assumes that the enzyme had a rigid and unchanging shape. So shown below is the induced fit model, and it states that the binding of the substrate to the enzyme induces a temporary change in the shape of the enzyme. The new enzyme better accommodates the shape of the substrate and a reaction occurs.
Like most chemical reactions, the rate of an enzyme catalase reaction increases as the temperature is raised. A 10 degrees centigrade rise in temperature will increase the activity of most enzymes by 50 to 100%. In the animation, we see the temperature start at 100 degrees. Slowly it is reduced to 45 degrees, then 5 degrees, and then 0. We can see the minimal movement at 0 degrees compared to the active movement at 100 degrees. Enzyme inhibition. An enzyme inhibitor stops the function of the active site, meaning the substrate can no longer bind to the enzyme. It can do this in one of two ways: competitive and non-competitive inhibition. Competitive inhibition involves an additional molecule binding to the enzyme in the active site, preventing the substrate from binding. In short, the molecule and the substrate are competing for the active spot. Non-competitive inhibition occurs when a molecule binds to the enzyme in a location other than the active site. The molecule interferes with the active site and causes a conformational change, resulting in the substrate and active site being unable to bind.
Now, let's explore two case studies: lysosomal storage disease in dogs and Fabry disease in humans.
Fabry disease in humans. Symptoms include kidney failure, pain, skin lesions, cloudy corneas and lenses, and heart disease. Fabry disease is a lysosomal storage disease which involves dysfunctional metabolisms of the spring phospholipids. Treatment includes pain management, dialysis, kidney transplantation, and enzyme replacement therapy, which is still very experimental. It's inherited on the X-linked chromosome. Another example is lysosomal storage disease in dogs. Symptoms include death, failure to thrive, balance issues, abnormal vision, blindness, fainting or seizures. This disease is caused due to a lack of enzymes needed to execute metabolic functions within the body. This leads to a buildup of substances in the body that would otherwise be expelled by the enzymes. There is no treatment available at this stage, and the transmission is a genetic inheritance.
Thank you.